Welcome to today's video where I will be looking at the cytoplasm and cellular organelles. Remember to like, share, and subscribe to my channel for more amazing videos. The cytoplasm of the cell is the jelly-like material formed by 80% of water. It contains a clear, liquid portion called cytosol, and various particles of different shape and size. These particles are proteins, carbohydrates, lipids, or electrolytes in nature. The cytoplasm also contains many organelles with distinct structure and function. The cytoplasm is made up of two zones. Ectoplasm is the peripheral part of cytoplasm, situated just beneath the cell membrane. Endoplasm is the inner part of cytoplasm, interposed between the ectoplasm and the nucleus. Cytoplasmic organelles are the cellular structures embedded in the cytoplasm. Organelles are considered as small organs of the cell. Some organelles are bound by limiting membrane and others do not have limiting membrane. Each organelle is having a definite structure and specific functions. Organelles with a limiting membrane include the endoplasmic reticulum, Golgi apparatus, lysosome, peroxisome, centrosome and centrioles, secretory vesicles, mitochondria, and the nucleus. Organelles without a limiting membrane include the ribosomes and cytoskeleton. The nucleus is the most prominent and the largest cellular organelle. The nucleus is present in all the cells in the body, except the red blood cells. The cells with nucleus are called eukaryotes and those without nucleus are known as prokaryotes. Most of the cells have only one nucleus and are termed as unenucleated cells, and a few types of cells have many nuclei and are termed as multinucleated cells. The nucleus is covered by a membrane called nuclear membrane and contains many components. The major components of nucleus are nucleoplasm, chromatin, and nucleolus. The nuclear membrane is double-layered and porous in nature. This allows the nucleoplasm to communicate with the cytoplasm. Nucleoplasm is a highly viscous fluid similar to cytoplasm present outside the nucleus. Nucleoplasm surrounds chromatin and nucleolus. Chromatin is a thread-like material made up of large molecules of DNA. Just before cell division, the chromatin condenses to form chromosome. Chromosome is the rod-shaped nuclear structure. It carries a complete blueprint of all the hereditary characteristics of that species. Nucleolus is a small, round granular structure of the nucleus. Each nucleus contains one or more nucleoli. The nucleolus contains RNA. Some functions of the nucleus include the control of all cell activities, such as metabolism, protein synthesis, growth, and reproduction. It is also involved in the synthesis of RNA. The nucleus sends genetic instructions to the cytoplasm for protein synthesis through messenger RNA or mRNA. It controls cell division and stores hereditary information in genes, ensuring the transformation of this information from one generation of the species to the next. The endoplasmic reticulum is a double membrane channel that is continuous with the nuclear membrane. It is involved in the intracellular exchange of material with the cytoplasm. Various products are transported from one part of the cell to another via the endoplasmic reticulum, making it crucial for intracellular transportation. Additionally, it serves as storage for synthesized molecules. Together with the Golgi complex, it acts as a synthesis and packaging center. The endoplasmic reticulum is divided into two types: granular endoplasmic reticulum, also known as rough ER, and a granular endoplasmic reticulum or smooth ER. Rough ER has a rough, bumpy, or bead-like appearance due to the attachment of granular ribosomes to its outer surface. It is mainly concerned with the synthesis of proteins in the cell, and also plays a role in the degradation of worn-out cytoplasmic organelles like mitochondria. On the other hand, smooth ER has a smooth appearance and is responsible for the synthesis of non-protein substances such as cholesterol and steroids. It is the major site for the storage and metabolism of calcium. Additionally, smooth ER is involved in the catabolism and detoxification of toxic substances, including some drugs and carcinogens. Ribosomes are organelles without a limiting membrane. They are composed of 35% proteins and 65% RNA. The RNA present in ribosomes is called ribosomal RNA, or rRNA. Ribosomes are essential for protein synthesis in the cell. Some ribosomes are found on the surface of the rough ER, while others float freely within the cytoplasm. Our protein molecules are subject to damage, and some cellular proteins may be needed for only a very short time. All such proteins must be destroyed, and this is where proteasomes come into play. A proteasome is a barrel-shaped organelle made of enzymes that cut protein molecules apart. Proteasomes are particularly important during cell division and embryonic development, periods when rapid and significant changes occur as cells become specialized. The Golgi apparatus is a series of flat membranous sacs, somewhat like a stack of saucers. Each Golgi apparatus consists of 5 to 8 flattened membranous sacs called the cisternae. It's present in all cells except red blood cells. The Golgi apparatus is situated near the nucleus. It has two ends or faces, namely the CIS face and the trans face. The CIS face is positioned near the endoplasmic reticulum, and the trans face is near the cell membrane. The major function is processing, packing, labeling, and delivery of proteins and other molecules like lipids to different parts of the cell. The processed substances make their exit from the Golgi apparatus through the trans face via exocytosis. The mitochondria are oval or spherical organelles bounded by a double membrane. The inner membrane has folds called cristae. Cristae contain many enzymes and other protein molecules that are involved in respiration and the synthesis of ATP. The outer membrane is smooth and encloses the contents of the mitochondria. It also contains various enzymes. Within the mitochondria, aerobic reactions of cell respiration take place. Therefore, mitochondria are the site of ATP production. Cells that require large amounts of ATP have many mitochondria to meet their need for energy. Other functions include the storage of calcium and detoxification of ammonia in the liver, and they are also involved in apoptosis. Lysosomes are single membrane structures that contain digestive or hydrolytic enzymes. When certain white blood cells engulf bacteria, the bacteria are digested and destroyed by these lysosomal enzymes. Worn-out cell parts and dead cells are also digested by these enzymes. This is a beneficial process and is necessary before tissue repair can begin. Lysosomes are often called the garbage system of the cell because of their degradation activity. About 50 different hydrolytic enzymes, known as acid hydroxylases, are present in the lysosomes. It is through these enzymes that lysosomes execute their functions. The hydroxylases include proteases, lipases, amylases, and nucleases. Centrosome is the membrane-bound cellular organelle situated almost in the center of the cell, close to the nucleus. It consists of two cylindrical structures called centrioles, which are made up of proteins. Centrioles are responsible for the movement of chromosomes during cell division. Secretory vesicles are the organelles with a limiting membrane and contain secretory substances. They are formed in the endoplasmic reticulum and are processed and packed in the Golgi apparatus. They are present throughout the cytoplasm. When necessary, these vesicles are ruptured and secretory substances are released into the cytoplasm. Cilia and flagella are mobile thread-like projections through the cell membrane. Cilia are shorter than flagella, and an individual cell has many of them on its free surface. The cilia beat in unison to sweep materials across the cell surface. The only human cell with a flagellum is the sperm cell. The flagellum provides motility to the sperm cell. Microvilli are folds of the cell membrane on the free surface of a cell. These folds greatly increase the surface area of the membrane. They are part of the cell's lining organs that absorb materials. The small intestine, for example, requires a large surface area for the absorption of nutrients, and many of its lining cells have microvilli. The cytoskeleton is the cellular organelle present throughout the cytoplasm. It determines the shape of the cell and gives support to the cell. It is also essential for cellular movements and the response of the cell to external stimuli. It consists of three major protein components. Microtubules are the straight, hollow, and tubular structures of the cytoskeleton, whose function is to determine cell shape and structural strength of the cell, and to allow the movement of granules to different parts of the cell. They form the spindle fibers which separate the chromosomes during mitosis. They are responsible for the movement of centrioles and structures like cilia. Intermediate filaments are the structures that form a network around the nucleus and extend to the periphery of the cell. Intermediate filaments help to maintain the shape of the cell. These filaments also connect adjacent cells through desmosomes. And finally, microfilaments are long and fine thread-like structures made of proteins called actin and myosin. They give structural strength to the cell and also provide resistance to the cell against pulling forces. They are responsible for cellular movements like contraction, gliding, and cytokinesis. Thank you for watching this video. Remember to like, share, and subscribe to my YouTube channel for more great content on anatomy and physiology.